Hi, my name is Jon Lorsch, and I'm the Director of the National Institute of General Medical Sciences at the NIH. And I'm here today to tell you about why you might want to consider spending part of your career doing scientific public service. And I want to start by telling you how I got to be at NIGMS. And it all started with this thing here, a cow's heart. When I was 4 years old, a teacher came to our classroom and brought with him a cow's heart. And while all the other students were probably screaming and maybe a few of them retching, I was fascinated by this thing with all of its chambers and flaps, and I decided on the spot that I was going to become a scientist and study biology. And that led to a whole series of chemistry sets and chemistry experiments done mixing things together on our kitchen table. My mother was a very patient woman. Eventually, that then led to some summer jobs working in labs in high school. When I got to college, I was lucky enough to work for these two people, Judy Voet and Nancy Hamlet. And in their lab, they were studying a new bacterium that Nancy had discovered from a sewage contaminated salt marsh, which was resistant to mercury. And my job was to purify the enzyme, the mercuric reductase, out of this bacterium that made it resistant to mercury. And through this work, I became fascinated with the molecular and atomic underpinnings of living systems, which is what I still continue to study in my lab today. Now, just as an aside, I'll point out for those of you who are struggling away in labs, wondering if your research results are ever going to be published or not, that this work with Nancy and Judy I started in 1986, and it was just published actually this year in 2013. There I am right there. So have faith if you're worrying about whether or not your research will ever come to fruition. Now, after college, I followed the career path that I had been planning for some time. I went to graduate school, I then went to a postdoc, and I was lucky enough to get a faculty position as an assistant professor. And from there, I climbed the academic ladder to associate professor and eventually professor with tenure. So this was the career path I had been hoping for and envisioned, where I was going to focus on research and spend most of my time in the lab. But actually, a funny thing happened on the way to that career path. Right around the time I started as an assistant professor, I was thrown into a small group room with a group of medical students and told I was supposed to teach them biochemistry. And they seemed like nice kids and very smart kids, and although I wasn't quite sure this was something I wanted to do, I had always thought I was going to focus on research, I decided I'd better try to do a good job. And so what I discovered was that if I tried hard to help them, they actually responded very well, and they learned the material, and not only did they learn the material, but they seemed to begin to enjoy it. And I discovered through this that I really had a love for teaching and for education, and this started a second career track for me in education. Through my work in education, that then got me involved increasingly in these three things: administration, management, and leadership. Because as I started out, I was just giving a few lectures, doing small group teaching, but soon I took on more responsibility and began running a block of the course, and eventually the whole course, and then eventually the first year curriculum, followed by becoming the chair of a number of committees that oversaw various aspects of education at the school. I was involved in a number of curriculum reform efforts. And now most faculty, if you talk to them, or at least some faculty, will tell you that these words are in fact bad words, that you do not want to become involved in these things, and the people who do are suspect. And that's kind of what I thought going into it. But what I discovered, the more I did leadership and management and administration, was that in fact, these weren't bad words at all, but they in fact are ways in which you can have a very big, hopefully positive, but potentially negative impact, if the wrong person's doing them, on a large number of people. And the more I did them, the more I realized that I could have a bigger impact on biomedical research and potentially society through leadership, management, and administration than I could just focusing on my own lab research. And so when the opportunity arose to apply for the job as director of NIGMS, I decided it was the most important thing I could do with my time. Now, let me just tell you a little bit about the National Institutes of Health. The National Institutes of Health is made up of 27 institutes and centers, shown here. Centers are smaller than institutes. National Institute of General Medical Sciences here is one of the institutes, and it's shown here next to the National Eye Institute. and the National Heart, Lung, and Blood Institute there. NIGMS is the fourth largest institute in terms of budget. We really have two missions, two overarching missions. The first is to promote fundamental research on living systems in order to lay the foundations for advances in disease diagnosis, treatment, and prevention. The second mission, which is related to the first, is to enable the development of the best trained, most innovative, and productive biomedical research workforce we possibly can. Now, I'll just highlight this phrase here, lay the foundation, because that really is fundamentally what we are doing. The research that we fund at universities across the country, the training that we fund at universities and schools across the country is designed to lay the foundation on which the other institutes, which are focused on diseases and organ systems, build their research efforts. NIGMS has 5 scientific divisions. They are the Division of Training, Workforce Development and Diversity, Cell Biology and Biophysics, Genetics and Developmental Biology, Biomedical Technology, Bioinformatics and Computational Biology, and finally Pharmacology, Physiology, and Biological Chemistry. Each of these divisions has a variety of program directors who work in it and manage grants in particular areas of science within those divisional boundaries. In 2014, NIGMS's budget was $2.36 billion, and this pie chart shows how we spent that money, how we invested that money. 89% of those funds went to support extramural research. So that's research that's conducted by professors, investigators all over the country at universities and other research institutions, so outside the walls of NIH. About 8% of the budget is invested in training and workforce development, again at universities and other schools across the country. A small fraction, about 3%, is required to run the institute, and a very small fraction, much less than 1%, is invested in a very small amount of research that goes on the NIH campus. And actually almost all of that is devoted to a single postdoctoral fellowship program called the Pratt program. So you can see that 97% of our budget goes to fund extramural research. Now, I just want to make the point that that $2.36 billion is not monopoly money. This is hard-earned taxpayer money, and we take that very seriously at NIGMS and the NIH as a whole. And our roles as good stewards of the taxpayer money are really critical. And fundamentally, what our job is to ensure that the taxpayers' money is invested in fundamental biomedical research in the most efficient and effective ways possible. So one of the things we've been focusing on for a long time at NIGMS is question-driven investigator-initiated research. Investigator-initiated research means that it's research which the investigators decide would be good to do. They're interested in certain questions. They propose those questions and strategies for how to solve them. Those proposals are reviewed by panels of experts who decide which are the best questions and the most likely to yield important answers, and then we fund the best of those, depending on how much funds we have available. The alternative is what we call programmatic research, in which some group advises the NIH that we should fund particular areas because of its importance for curing a certain disease, for example, and therefore we put investments in those targeted areas of science. Now, why do we focus so much on investigator-initiated research? And it's because ultimately, investigator-initiated research saves lives. And let me just give you a few examples of what I mean by that. So, for example, studies of virus-resistant bacteria led directly to the discovery of restriction enzymes. Restriction enzymes cut up DNA and allow us to put it together in different ways. And the discovery of restriction enzymes led directly to the development of the entire biotechnology industry. And the biotechnology industry, as you may know, produces a wide variety of drugs and other important materials. So, for example, production of insulin for use medically in humans, or human growth hormone, or antibodies for medical use, all depended on the discovery of restriction enzymes. Similarly, studies of bacterial DNA replication led directly to the discovery of the anti-cancer drug cisplatin, which has saved hundreds of human lives. Studies of proteases and DNA polymerases, enzymes, were essential to the development of anti-HIV drugs. And finally, studies of the enzymes involved in cholesterol biosynthesis and the signaling pathways that control these enzymes were essential for the development of the statin drugs for controlling cholesterol levels in humans. So why do we need to focus on investigator-initiated research? We are trying to bolster our support for it, and it's because over the years that support has eroded for a variety of reasons. So what this chart shows on the y-axis here is NIGMS funds that were invested in targeted research. So that's that programmatic research I told you about that's directed to specific scientific areas. And that's these blue bars here. And you can see that in the early 1990s, a very small fraction, a small amount of NIGMS's budget was invested in targeted research. Between 1998 here and 2003 here, you can see, however, it grew dramatically. And those were the years of the NIH budget doubling. And it made a lot of sense when there was always new money coming into the system during the doubling to invest some of it in these targeted research areas to try to catalyze new areas of research, try experiments in new ways of organizing research, for example. Now this red line here, which is this other axis, shows the percent of our grant portfolio that was invested in investigator-initiated research. And you can see again, it's sort of the reciprocal. In the early 1990s, 99% of our portfolio was invested in investigator-initiated research. Again, during the budget doubling, this fell to the point today where it is just about 80%. So we went from 99% investigator-initiated to 80% investigator-initiated. Now that the budget doubling is over, it seems quite important that we re-equilibrate the system and bring this red line back up to increase the amount of investigator-initiated research we support, and that's going to require bringing these blue bars down. Now, a couple of other important questions that we're focusing on right now at NIGMS. The first is, can we develop more stable, flexible, and efficient ways to fund this question-driven investigator-initiated research? In the current system, in which we fund lots of smaller projects, each investigator breaks his or her research up into a number of different projects. That is a fairly unstable system because they could lose these grants fairly easily. It's not flexible because the projects are very defined and prescribed in advance. And so we think that that may be dampening investigators' willingness to take risks, to be creative, and in general, making the impact or the ambitiousness of their science not quite as high as we would like it to be. And so we're looking for experimental ways that we can see if we can develop more stable, flexible mechanisms, hoping that this will increase the creativity, the willingness to take risks, and the ambitiousness, and thus give taxpayers a bigger bang for their buck. We also think that such mechanisms will be more efficient in terms of allowing us to fund more science for taxpayers' money. We also are asking how we can promote innovation and experimentation in the training arena to find the best models for biomedical education and training. And so you may say, well, we've been training scientists for decades. Why do we need to find new models? Aren't the ones we have perfectly good? Well, let me show you something here. So this is a graph I got from this paper in Biochemical Journal. And what it shows on this axis here is basically biomedical information. They call it percentage volume, which is the percent total defined as what it was in 2010. But you can think of it as biomedical information. And it's told in several different lines. The red line here is publications deposited in PubMed, the database. The blue line here is structures of proteins and other macromolecules deposited in the protein data bank. And the other lines there are sequence databases and how many structures are deposited there. And what you can see in all these curves is that before about, say, 1990, knowledge was growing linearly to some degree. After sometime around 1990, you can see it takes off like a rocket and grows exponentially. All these curves tell the same story. And so at least the amount of information and therefore at some level knowledge we had changed dramatically in the last two and a half decades. And I think you will agree that many other things in science have changed as well in that time period. The complexity of what we know the interconnectedness, the need for interdisciplinary research, etc. Now, if we put another axis on this graph, which is some measure of the biomedical graduate education model being used, that is the way in which we teach graduate students, it would tell a very different story. So this graduate education metric would look like this orange line that is completely flat. So to a first approximation, since 1990, when I started graduate school, in fact, very little has changed in how we educate graduate students. And I think the difference in shapes of these two sets of lines, the graduate education model and the biomedical knowledge model, indicate that there may be some disconnect. Things have changed dramatically in science, and they have not changed dramatically in how we educate scientists. And so I think it's possible, at least, that we're not using the optimal models right now to educate the next generation of scientists, and we should therefore do some experiments to try to find what the optimal models are in this new regime. Now, just to end, if I have done my job and convinced you that you should get involved somehow in scientific public service, let me tell you how you might do that. So if you're a trainee, a student or a postdoc in a lab, or a researcher working full-time in a lab, that doesn't mean you can't get involved in some way in public service and science. You can, in particular, through the professional societies, find a wide variety of opportunities to volunteer some of your time to get engaged in making the biomedical research process more efficient and more effective. If you'd like to follow a full-time track in this regard, there are also a variety of ways you can do that. One, again, is through the professional societies, which have a number of jobs that require scientific training to do. You can also work in foundations that fund biomedical research and help promote it in other ways. If you'd like to take the government track, as I have, there are a variety of possibilities. For instance, program directors who manage grant portfolios, as I mentioned, and promote science in different areas in a variety of ways. Scientific review officers are essential scientists who run the reviews of grant applications and make sure that the best ones are the ones selected for funding. Program analysts do a variety of tasks, including evaluating our programs and seeing how effective they are and helping us optimize them if they aren't working as well as we'd like them to. And finally, a variety of communications fields, such as writing, is required in order to tell the public what the science is that's been discovered. And lastly, if this is a track you'd like to explore, a couple of resources are the Presidential Management Fellowships and the AAAS Public Policy Fellowships, which are very competitive, but if you do get one, they're excellent ways to get experience in scientific public service. Thanks for watching. I hope to come back sometime.